Right, thank you very much. And I too would like to echo my pleasure in being here to speak to you today. I think it's fascinating, the theme in our slides, because I too didn't plan this slide knowing my last speaker's last slide, but this is a bit of my theme as well. So today I'd like to tell you about my passion, my research in prion disease, and how a twisted protein can kill your brain. So what are prion diseases? Well, the technical name for them is transmissible spongiform encephalopathy, which is why we say prions, because that's a bit of a mouthful. And this includes a number of different diseases. In humans, it can cause the following diseases. It affects sheep in a disease called scrapie. Of course, as you heard, it affects cattle in the disease we know as mad cow disease. And maybe some of you have heard of chronic wasting disease. It affects deer, elk, and moose. And I'll talk a bit more about that disease later. And fortunately for me as a researcher, it also affects rodents, because this is the mainstay of some of the way we do our research. So in humans, this disease is always fatal. Early on, people come with difficulty with coordination, some trouble thinking, and as they progress, they stop being able to talk, they stop being able to move until they die. And any idea how long this disease takes? The average time to death after symptoms starts?
1 year.
4 months. For the most common form of this disease, 4 months. So this is a very fast disease. It's what we call a rapidly progressive dementia. So what causes this? As Jay mentioned, these are caused by prions. Well, what is a prion? The interesting thing about these diseases, they can happen spontaneously. In fact, 90% of cases are spontaneous, not related to eating contaminated food, anything like that. But you can also be infected with this from surgery or blood transfusions. And not only that, you can inherit this disease. So a very unusual type of disease. And the infectious agent itself is really difficult to kill, unlike typical viruses or bacteria. So what makes prions unique? Well, if you think of a virus normally will infect a cell, inject its DNA or RNA, replicate that, make new viruses, and go on with its life cycle. Well, a prion does the same thing. It infects a cell, replicates itself, moves on, but it's only protein. So how on earth does that work? That's the fascinating bit about prions. Well, in order to explain what we know, I have to introduce you to a protein that I spend a lot of time with. It's called the prion protein. Now, protein isn't just what you add to milkshakes at the store. Proteins are actually molecules that are made inside our body and do many, many different functions. We couldn't survive without proteins. This particular protein lives on the surface of our cells. It lives on cells in our brain, in our heart, in fact, all over our body. And you can see we know a lot about this protein. This is actually what it looks like. This is its shape. You can see I've got all these labels on it, and it's not actually pink and yellow, but the shape is correct. And the shape is really important because proteins function based on shape. So the problem in prion disease is this protein changes shape. Now you notice I don't have a bunch of diagrams and labels because it doesn't actually turn into a bunch of pink triangles. But we don't actually know the shape, but it does change. And the problem is once it changes shape and it sidles up to a normal prion protein, it makes it change shape. And then this process continues and you get a buildup of these misfolded proteins. So shape is really important. And that's what I'd like to talk about in sort of the first project that my lab's been involved with. Some of you may have seen this article, it was last year in March, which talked a bit about our research and the importance of shape, and not just shape, but shape shifting. So if you imagine the prion protein doesn't just form pink triangles, but it can form any of these shapes. All right, squares, diamonds, any array of shapes, or what I would call a cloud of different shapes. Now, one particular species can form a cloud that has red squares and blue diamonds and green triangles. A different species, that prion protein, might be able to form the other shapes. So if you take one pink square and square, diamond, and triangle from species 1 and try to infect species 2. It's not going to work. The shapes don't match. Aha, but what happens if it encounters another species whose cloud overlaps species 1? Well, now I think you can see you can pass from species 1 to 3. Well, let's introduce another species, and I think you see how it can progress from one species through to the final species. Now, this isn't necessarily only the case for between different species, but it can actually happen within species as well. And that's sort of what we found, and I don't seem to be able to advance the slide right now, so I'll let you figure that out. But this can happen between, say, a different animal, such as in chronic wasting disease, deer or elk or caribou, and could that then transfer into humans by going through an intermediate host? And this is a very serious concern we have right now. So another project that we're looking at, so in addition to just shape, is if you imagine you have all these red triangles or red shapes, how on earth do you get from that to actually causing a person to die in 4 months? You know, what's happening there? And we know that these proteins, these shapes accumulate in the brain, and then the brain itself develops holes, full of holes. In fact, that's why the disease is called spongiform encephalopathy, because it starts to look like a sponge. So something we're interested in looking at is the brain itself and what happens during disease. To study this, do I have hope? No? Okay, well, I will pretend I'm a brain with holes in it. I should have brought my fiddle, I knew it. Anyway. So basically, you develop holes in the brain, but it's a bit difficult to say, look at the brain as it is without some help. I saw a brief glimpse there. Oh, so, okay, I'll quickly run with this as it's going. There it is. All right, I'll go back one slide. This particular approach that I have, we call prion disease in a dish. And basically, we take an animal, a mouse, we use its brain, sort of the back part of the brain, we slice it up, and we actually plate it in a dish. This is starting to sound like food again, but we don't actually eat this. Here we can actually grow the piece of brain. For weeks and weeks and weeks, we can infect it with the prions, this infectious material, and it will actually, we can monitor it over time and see if it develops disease. So when we look at this, this is the actual brain slice now that's been growing in a dish for 35 up to 49 days. And we've colored it, the different cells. So a lot of the neurons are stained white, and another special type of neuron is stained green. And they're very healthy looking. You see, over time, everybody's doing well. And this is an uninfected slice. Well, compare that now to an infected slice. And I hope you can appreciate that with time, we lose those neurons, those nerve cells. The white signal is almost completely gone. Now, if we look more closely at an area in the infected slice, the green area, we can look at one of these green cells. Oh, it's freezing again.
Technology's failing.
Yeah, it's all right. It's the only problem with images, because I don't know if I can pretend that I'm a big neuron. But on these big green neurons, there's a bunch of little spines, so these little processes, because in order for a neuron to communicate with other neurons, you imagine my hand and my fingers, it has to talk to my other hand and fingers. And these fingers are important for communication. These fingers are important for how neurons talk to one another. And what we see in the animal is those fingers are lost as the disease progresses. And what we found in our slices is that when we watched for those signs as well, we also saw that those fingers were lost. There they are. Quick. All right. So these are the little fingers that I was talking about. And then in disease, they vanish, they disappear. And the good thing about this is it really means that this model, it's not just something growing in a dish that can sort of make prions, but this is actually replicating some of the disease. And it does it faster than we see in the animal. I'm talking about 40 to 50 days here. In some animal models, we can be talking for hundreds of days, potentially years, to see the same things. Plus, it's in a dish, so it's a lot easier to study and to look at. So what we have funding from AI Bio is to apply this technique to a bunch of different situations. So specifically, we're interested in as much as we care for the mice, we're interested in other species and other strains. So we've actually started looking at chronic wasting disease. And here's just a couple pictures. This is actually after 70 days of culture. And using low dosages of the prions to try to infect the slices, they actually stay quite healthy, as you can see by all the white staining. But if we use a higher dose, by 70 days, they're quite unhealthy and they're starting to also go through the disease process. The little bit of red you see as well is a sign of active dying cells. We're also applying this to other parts of the brain. As I showed you, we're just using the back part of the brain right now. Well, all of the brain can be affected by prions, and different shapes of prions affect different parts of the brain. So we want to study that. So we've started culturing and infecting other areas of the brain. And this is sort of an area sliced like this through the brain. And again, showing nice healthy cells over time. And we've been able to infect these as well. Finally, it also gives us a place to test therapies a lot faster than in an animal or combinations of therapies. And so if I look at a slice here that's been infected, that's not treated, that's looking unhappy, as my technician would say, once we treat it, we start to see a bit of rescue. So some of those neurons are surviving now. And so this gives us a way to screen and hopefully advance treatment ideas for these diseases. So you may say, well, why? Why should we worry about this? Is mad cow, is that around anymore? Is it a problem? Of course, as a clinician, I care a lot about the humans that still get this disease. But I think you should also be aware of chronic wasting disease. Because in Alberta, it first appeared in a farm in 2002 in white-tailed deer. By 2005, it was in wild mule deer. By 2012, it was in wild moose. And if you look at a map of how this is progressing, how this is spreading, in 2007, this sort of gives you a North American view of chronic wasting disease. I'd like you to pay attention to the gray squares, the light gray squares. That's all of the chronic wasting disease in wild, free-ranging populations. And I'll show you over time what's been happening. And this map now is current as of April 2004. And I'd like you to note in particular Alberta. So this is a growing and rising concern. We don't know all of the implications yet to human health, to tourism, to the industry, but it's certainly not going away. So with that, I would like to thank all my collaborators, my funding agencies, and for your attention.